Sir,
Yeah.
sir, now you can start the class, sir.
Okay. Can I please know before that, is there anybody waiting at 8 o'clock for another class?
I didn't get you, sir.
Is there another class scheduled at 8 o'clock and somebody is going to wait for 8 o'clock class?
Yeah, sir, we have another class as well, sir.
Yeah, what time? 8 o'clock.
After 8, sir, 8 p.m. we have. But you
Okay.
can carry on, sir. If it is too long, means sir, it's okay, sir. We'll take this class, sir.
Okay.
Okay, sir.
Okay. Good evening. My name is Dr. Shakti, and we'll be starting with hematology now. So when we talk about hematology, one of the main important question that is going to come from hematology is your erythropoiesis. Okay, so starting off with what is the definition? So the thing is, this question comes as an essay by itself. So this is very important. Out of all the topics in hematology, this is arguably one of the most important topics. Apart from that, platelet is also another topic. So these two topics usually come as an essay by itself and often repeated steps in erythropoiesis, factors affecting erythropoiesis. Okay, so start off with what is the definition of erythropoiesis? That is pretty simple and basic. That is just the process of formation of new RBC. How long does it normally take? Around 5 to 7 days. Okay, so while erythropoiesis is happening, usually you have a terminology called erythron. So erythron is actually, you know, it's a pool, a dynamic changing pool, where the precursors are there. Along with that, mature circulating RBCs are also there. Okay, so usually, right in the center, there will be a macrophage and surrounding it around 30 to 40 different precursor and mature cells, full mass of them is there. Okay, that we call it as erythron. Now, that is the first question, definition. Second question, what is the site of erythropoiesis? So site of erythropoiesis, starting in the intrauterine life, to a child and in the adult stage. These are the three headings under which you have to talk about this. So in the intrauterine life, there are three phases. In the first 3 months, we have the mesoblastic phase, where from the yolk sacs, mesoderm, that is where your erythropoiesis is taking place. So this is from the yolk sac it starts. And then it starts going to the liver and the spleen. So then comes the hepatic phase in the next 3 to 5 months. So first 3 months is mesoblastic, mesoderm of yolk sac. Then we have 3 to 5 months liver, spleen. Okay, so that is your hepatic stage. And lastly, beyond that, more than 5 months, liver, spleen, along with your bone marrow of all the bones. So all the bone marrow, all the bones are involved over here. So this is in the intrauterine life. Once the child is born, slowly it starts shifting till the age of puberty is achieved around 18 to 20 years. Usually all bones that have red marrow. Okay, so it's not basically any bone, but only those with red marrow. Along with that, to a degree, liver and spleen is also involved in erythropoiesis in children. On adults, you see that it is only in the red bone marrow. So where exactly is the red bone marrow? You just have to think about your body right in the center. Okay, the skull, the vertebra, the ribs, sternum, pelvis, scapula, these are all located right in the center of your body and the ends of long bones. So that is the second question. So definition, and then comes the size of erythropoiesis. Then what are the steps in erythropoiesis is the next question. So steps of erythropoiesis can be divided into two stages. First is the engagement phase. In the engagement phase, initially we have the pluripotent, uncommitted hematopoietic cells. These are not committed to anything yet. They are uncommitted as of now. So this uncommitted hematopoietic stem cell starts to become committed. Okay, so we have uncommitted hematopoietic cell that can essentially become any type of hematopoietic cell. That has to be committed into your erythropoiesis. That is your engagement phase. So it gets committed to a hematopoietic stem cell, which is committed, and it goes into a myeloid stage. There are two stages, either myeloid or lymphoid it can go. Here, it has to go into your RBC. So first it goes into a myeloid stage, and from the myeloid stage, it forms a BFU, that is burst forming unit erythron, which ultimately goes to a colony forming unit erythron. So this is your engagement phase that we're talking about. So pluripotent uncommitted to become committed, we have GM-CSF, G-CSF, SCF, stem cell factor. Okay, then interleukins like 163. Then from committed to become the myeloid precursor, GM-CSF and interleukin 3 are involved. And again, interleukin 3 will help it convert to finally form the burst forming unit. And this is where your erythropoietin is now going to help convert it into colony forming unit. So this is the engagement phase where CFUE is formed. Now from here on, it is going to go through various phases that is exclusive for your RBC. So the colony forming unit, hemocytoblast, that becomes the proerythroblast. Proerythroblast becomes basophilic erythroblast. Basophilic becomes polychromatic. Polychromatic becomes orthochromatic. And finally, a reticulocyte is formed. So in the stages, initially what is happening is the entire cell is going to be blue in color. Okay. Eventually, hemoglobin is going to come in. So now the blue color, because of the RNA elements, ribosomes and all that. Along with that, hemoglobin is starting to appear now. So that is your polychromatic where two different colors are existing. Then from there, your orthochromatic stage is there. So as the hemoglobin starts to build up, it will go inside the nucleus, go to the histone, inactivate it, resulting in your changes in the nucleus. And eventually that will get extruded out. So we have the reticulocyte, which is devoid of any nucleus. Okay. And reticulocyte still has some remnant fragments of RNA, ribosome, and all that. So this reticulocyte now stays inside, and in 1 to 2 days of maturation, it is released into the circulation. Okay, where during this maturation phase that is happening, that is where all of these elements which are there inside the cell is disappearing to finally form your mature RBC. So this is a very important stage where the final maturation of the RBC is taking place. Okay. So these steps are what are called the terminal differentiation. So initially we had the engagement phase. Now we're having the terminal differentiation. So in terminal differentiation, there are a few things that happen, which is pretty common to all the stages. So while it is going on, one thing, hemoglobin is slowly building up. So gradual accumulation of hemoglobin. Two, the size of the cell decreases. It starts from around, 20, okay, around 20 nanometer. From there, it goes to close to around 7.5 in RBC. Okay, in reticulocyte, slightly more 8. Okay, so from 20, even before the colony forming unit can be 35. From there, the size decreases and the nucleus also condenses. So these are various things that happen during terminal differentiation. Often questions come from this as to when is hemoglobin first seen. So when is hemoglobin first seen? That is polychromatic erythroblast. This phase over here, polychromatic erythroblast, is where you first see hemoglobin. But when is the production of hemoglobin happening? That starts right from the proerythroblast, actually. It's just that the stains that we normally use cannot show it. Okay. But actually, the production of hemoglobin starting somewhere in the proerythroblast. But it starts appearing in the polychromatic erythroblast phase. Okay. So these are the various stages. You can see the proerythroblast, that becoming basophilic blue in color. mix of red and blue, that is polychromatic. Then, as you can see, the nucleus is becoming condensed, almost getting pycnotic, and it's extruded out. In reticulocyte, you still have some remnants which are still there. And finally, we have the mature erythrocyte. So this is one terminology that is there. Apart from that, you can even call these as normoblast. So a pro-normoblast becomes a early normoblast, intermediate normoblast, late normoblast. Or if you're using the terminology erythroblast, a pro-erythroblast becomes basophilic, polychromatic, orthochromatic. Okay, if it's normoblast is the term that you're using, you can describe it as a early, intermediate, and late normoblast. So you can see here the basophilic erythroblast. This has something called a wheel spoke nucleus usually. Hemoglobin starts appearing and it forms a polychromatic. Now the hemoglobin goes inside the nucleus, as I was explaining earlier, resulting in chromatic inactivation and nuclear condensation, and eventually it forms your orthochromatic. This has somewhat like a cartwheel-shaped appearance. And then this nucleus is extruded out, resulting in your reticulocyte, which has some remnants. So these remnants include your ribosome and mitochondria, some parts of Golgi and cytoplasmic vesicles and all. Now, it stays in the bone marrow for 1 to 2 days and 24 hours it stays in circulation, and eventually it will get sequestrated in the spleen, where a series of changes happens. And finally, a mature RBC is formed. So your mature RBC usually has a range of 6.5 to 8.5 micrometer. Okay, it is biconcave. It has a volume of 78 to 94 femtoliter, lifespan of around 100 to 120 days. Okay, so this is a microscopic diagram of your reticulocyte with the fragments that you can see, and these are all the mature RBCs that you see. Again, can you see how BFUE, CFUE, proerythroblast, basophilic, it starts becoming polychromatic, orthochromatic, reticulocyte, and you can see that the size is decreasing, nuclear condensation is happening until it is extruded. And here also you can see from proerythroblast, hemoglobin synthesis is actually starting. Okay, it's just that over here it starts becoming visible. These are again another group of diagrams for the same thing. Okay, so with diagram, explain the whole process, please, because it becomes much easier for the examiners and you are bound to fetch more marks out of it. Next is understanding what is erythropoietin. So under erythropoietin, we'll talk about what is the structure of erythropoietin, where is it produced from, hypoxia-inducible factors, actions of how does erythropoietin act, what are the factors affecting erythropoietin. Okay, when you talk about erythropoietin is a glycoprotein. So it's a protein. Okay, what is it? A glycoprotein. So around 74% of that is protein, 25% of that is carbohydrate. Where is the source of production? Majority of it is produced in the kidney. Okay, right next to the nephron. So in the nephron, peritubular capillary bed, there are certain interstitial cells. So there are interstitial cells, just peritubular capillary. So there is a capillary which is there, that peritubular capillary. Next to it, there is a interstitial cell. So majority of the erythropoietin is produced from this kidney. Remaining around 15% is produced by hepatocyte, perivenous hepatocyte. Okay, so we have the kidney and liver. Apart from that, erythropoietin is also a local hormone which is there in all over the body. So just a couple of examples: one in brain, one in uterus oviduct, which has its own activity over there. Okay, but the main source of circulating erythropoietin, that is from the kidney and liver. Rest of it are local erythropoietin which are acting at local level. Okay. So how does erythropoietin release happen? One, hypoxia, lack of oxygen. So when there is lack of oxygen, PeO2 is less. That is going to stimulate something called hypoxia-inducible factors. It's a transcription factor. Okay, so when hypoxia-inducible factors are activated, there is actually HIF alpha and beta. Okay, so this HIF1 has alpha and beta, and that gets activated and goes into the nucleus to a particular area, which is called hypoxia response element. This is right next to the erythropoietin gene. It's not only erythropoietin which is released from here, there are a group of other things which are released, which are all made in order to tackle your hypoxia, which stimulated this. So initially there was hypoxia. That resulted in hypoxia-inducible factors, which are going to activate your hypoxia response elements, which will in turn result in gene activation of erythropoietin gene. One thing to counteract hypoxia, produce more RBC so that more oxygen is made available. Two, create new blood vessels, angiogenesis. So for that, vascular endothelial growth factors are released. Again, you need vasodilation to make sure that the blood flow is adequate all over. Okay, so for that, nitric oxide is formed. Nitric oxide synthase is formed, from which nitric oxide is formed for vasodilation of the capillary beds. Now, beyond that, another problem is hypoxia. If oxygen is not available, you try to make sure that anaerobic metabolism can at least sustain the person. Okay, so glycolytic enzymes are also increased. So these are all the things that happen when hypoxia is happening. All right. So just for those of you who need added information, okay, who want to make it a little more thorough, this hypoxia-inducible factors, usually there's normally when oxygen is adequately there, there is a particular enzyme called proline hydroxylase. This proline hydroxylase requires oxygen. So in the presence of that, this HIF binds to something called VHL, on-hypolindryl protein. Once it is bound to that, this tags it for degradation through a ubiquitin and proteasome degradation pathway. Okay, so normally what happens? HIF is degraded because of the presence of a proline hydroxylase, which requires oxygen. When there is hypoxia, proline hydroxylase cannot act. So instead, the HIF alpha is free. Now, this HIF alpha will go and combine to another component which is called HIF beta. Okay, so HIF alpha and HIF beta, both of them will combine now, go into the nucleus and attach to a CREB, cyclic AMP response element binding. Okay, so that is also an area. And along with that, it goes to hypoxia response element. Okay, so remaining is whatever we already talked about. Now, erythropoietin is important especially in two stages, early stages, BFU to CFU. Okay, and CFU to proerythroblast. These are two stages where your erythropoietin is important. So, what are the functions of erythropoietin? One, early differentiation, what we just talked about. Okay, BFUE, CFUE, pronorthroblast. So the thing is, normally we were seeing a time range of around 5 to 7 days. So instead of 5 to 7 days, now this whole process will take 2 to 3 days. Okay, so this is very important that it speeds up the process of early differentiation. Not only early differentiation, almost every step in erythropoiesis is made faster, especially early differentiation. But beyond that, every step in erythropoiesis is made faster. Okay, along with that, it also increases hemoglobin synthesis. And whatever is formed, that reticulocyte is being released also faster. And the RBCs which are there, it inhibits apoptosis of that. So in a way, what happens? The early differentiations increase, all the steps in erythropoiesis are increased, increased hemoglobin synthesis is there, whatever reticulocyte is finally formed is being released, and whatever RBC is outside, its apoptosis is inhibited so that it can last longer. So erythropoietin does all of these things. Now, a very important question: factors affecting erythropoiesis. So this is one question that is, repeated countless times. If you don't go to the university exam without studying this, you might as well just fail. So this is that important a question. So what are the factors affecting erythropoiesis? Two things we have to talk about: general factors, special factors. Under general factors, you can talk about hypoxia, vasoconstrictor agents, nucleotides, products of RBC destruction. Beyond that, you can talk about special factors: proteins, minerals, vitamins, cytokines, hormones. Okay, so we'll go to it one by one. One, hypoxia. So hypoxia, as we're talking about, is one of the most potent stimulus. So under this, you can talk about, how hypoxia inducible factors, hypoxia response element, erythropoietin gene, what are the things that happens. All of this you talk about, whatever we already discussed, talk about it over here. Okay. Apart from that, certain substances like catecholamines, which result in a lot of vasoconstriction, that in turn causes a bit of hypoxia. So vasoconstrictor agents, presence of nucleotides like cyclic AMP, NAD, and ADP. This is important because, when your ATP is being utilized in your system, so oxygen is required for that. Now, because of metabolism and not enough oxygen is available, in such a scenario, you have a lot of nucleotides which are available. So that indirectly is telling you, okay, hypoxia is there. Okay, or in any way, whatever RBCs are there getting destroyed a lot, and products of RBC destruction, such as hemosilate. So ultimately, what is it telling you? Okay, either there is hypoxia in the body, or a lot of RBCs are getting destroyed. So one, hypoxia. Two, vasoconstricting agents. Three, nucleotides. Four, products of RBC destruction like hemosilate. Okay, so these are the general factors. Apart from that, the special factors. In special factors, you'll have to talk about the different nutritional requirements which are required for erythropoiesis. The major ones, protein. Why protein? Because if you know the structure of a hemoglobin has a globin chain, which is basically made up of amino acids. For that, you require protein. Okay, even in your general plasma membrane is 50-55% protein structure. Okay, so proteins are essential over here. Two, your vitamin B12, cobalamin. Okay, and your folic acid. So both are required for DNA synthesis maturation and all. Okay, then iron is very essential because it forms an integral part of your hemoglobin. Okay, it's in fact called iron protoporphyrin 9. Then we have certain other factors like vitamin B6, B2, C, E. Okay, at least the ones I marked in yellow, you have to write it. No. Then you go to the blue ones, vitamin B6, heme synthesis. Vitamin B2, oxidative reactions. Vitamin C is involved in iron metabolism because it acts as a reducing agent, retaining iron in the ferrous form. Okay, and vitamin E, again, protection against oxidative. Okay, and then we have copper also, which is also required for the transport of iron. Now, these two things that we're talking about, folic acid and vitamin B12, which are very important for erythropoiesis. Folic acid is usually found in green leafy vegetables and all. On the other hand, in meaty products, milk, okay, red meat, fish, egg yolk, milk, all of that is where vitamin B12 is found. Okay, so in vegetarians, generally vitamin B12 can go down. Okay, now this vitamin B12 is stored in liver and it can exist for a long time. So there should be a prolonged period of deficiency for it to actually manifest. Folic acid, on the other hand, its reserves run out in a few months. Okay, and especially, certain situations like pregnancy increase its requirement also. So imagine if its reserve is less and now the person gets pregnant and its requirement is increasing. Okay, that is where you have to understand that folic acid will manifest much faster, the deficiency of that, than vitamin B12. So where is this important again? There is a particular thymidylate synthesis. So ultimately, what does it do? You learn this in biochemistry. Okay, the uracil to thymine. So in this conversion in DNA synthesis, this part of folic acid becomes a methylene donor. Okay. Apart from that, the hormones which are there, erythropoietin, we already talked about it, how it helps and all that. You can talk about that. Okay. Then cortisol, thyroxine also increases through increasing BMR. The requirement for oxygen is increased. Okay. These are two things that you have to know. Testosterone and estrogen. Testosterone, what it does is it increases the sensitivity of erythropoietin to the target cell. So whatever erythropoietin is released, it goes and acts somewhere. So those cells become more sensitive to erythropoietin. Two, estrogen. Estrogen, what it does is one, it decreases the erythropoietic response to hypoxia. Okay, it also decreases the synthesis of the globin chain. These are two essential ways by which testosterone and estrogen act. And that is why the difference between males and females with regard to the RBC count actually. Okay. And then we have cytokines we had initially talked about, interleukin 163, GM-CSF, GCSF, SCF. Okay. So again, these are the various things we have to talk about. This is a very, very, very important topic. Okay. So we just talk about why I choose Auricle. Auricle has all of these various perks which are available. One, that is recorded classes, and these pre-recorded classes are made available to you at your own convenience. You can read it. We have live classes which are there, like the one you are, looking at right now. And we have express exam module which is there. So, you can complete the syllabus in less than 20 hours actually. This is very essential if it's right before the exam. Okay. And then one doubt clearing sessions are there, hard copy note sets are there, and different types of questions are there as an MCQ pattern, because that will be helpful for your university as well as in the future for your NEET, PG, and FMG and all. Practical sessions are also recorded and classes of that are also present. So these are the various subscription plans which are available. You can use my code Dr. Shakthinag 20 for discount on subscription. Okay, so you can follow the different social media handles of Auricle in case you want to reach out to us. So again, thanks for watching. See you again next time.